Today's lesson is topic 6.4, translation. Our learning objective is to explain how the phenotype of an organism is determined by its genotype. So let's look big picture here. So our same central dogma diagram that we had in 6.3. Your genotype, your genes. All of your genes are carried on your DNA. That information then is transcribed to RNA, and RNA then is translated to protein. And when it comes to proteins, proteins are what give us a visible phenotype. So transcription is going to transcribe a gene to make our protein, which gives you your phenotype. Oftentimes we like to think of it as one gene makes one protein, which makes one trait, and generally that's true. There are exceptions though. We think about the hemoglobin protein. Most people have normal hemoglobin that carries their oxygen fine. However, some people have sickle cell disease. That's their phenotype. What that means is that their hemoglobin is misshapen. It's the wrong protein. And so with hemoglobin, there are multiple polypeptide strands involved. So each gene codes for one of those polypeptides that are then folded together to make the hemoglobin protein. And someone who has sickle cell trait or sickle cell disease has a problem with their hemoglobin protein. It's not folded correctly, and that's due to a substitution, a change in one base that codes for a different amino acid, changing the whole shape of our protein. So today our focus is going to be on translation. The second step, how we go from RNA to a protein. So translation, mRNA to protein. We did transcription in 6.3. So translation of mRNA to a protein occurs on the ribosome in both prokaryotic and eukaryotic cells. This process translation is the same. All living things have the same genetic code. That helps to provide evidence for common ancestry. And we'll get more into common ancestry when we talk about natural selection and evolution later on. But all living things have the same genetic code. We can take a gene from a jellyfish and stick it into a bacteria and have that bacteria express the gene. Or another common example, the insulin gene in humans. We have taken that gene, just the gene for insulin, put it into bacteria, and now the bacteria produce insulin that then can be used by diabetics. And so most insulin that's used in diabetic patients is made by bacteria, because they're just going through protein synthesis with the embedded or inserted insulin gene. Now in 6.3, I had mentioned retroviruses. Oops, there we go. Too far. Too many buttons. With this reverse transcription, going from RNA to DNA. There we go. So our retroviruses are RNA viruses, which means that they carry RNA as their genetic code. They go through a reverse transcription process to make DNA from an RNA. And that's how they integrate with their host when they attack. Some examples of that include HIV and rabies. And then we'll talk about this more later. We'll revisit viruses. So some vocabulary terms to know. We have transfer RNA. This transfers amino acids to the ribosome. Each tRNA codes for a specific amino acid. So on one end of our tRNA, and this is just one single strand of RNA that has folded up on itself. At one end, we have the anticodon. The other end would be our amino acid. And so our anticodon We've got 3 nucleotides here making up our anticodon. This pair is complementary to the codon on the mRNA. So here's our mRNA, triplet code, so 3 nucleotides. This is going to code for a specific amino acid. So here we have GAG. And here we have a codon chart. This is how scientists have figured out how to read our genetic code. If we look, here's our codon, GAG, codes for this amino acid. This is our universal genetic code. This is consistent throughout every species, all of these 3 codons, and what amino acid they code for. Again, showing some relatedness. We all have the same genetic code. Right, our next term, wobble. So in the code is not always as specific as one codon for one amino acid. We look here for serine. There are 4 amino acids, all coding for serine. Wobble refers to this 3rd nucleotide in our codon. And so this 3rd base of our codon and the corresponding base of our tRNA are not as strict. So if there was, say, a typo in this mRNA, a mistake happened during transcription that didn't get caught, then it's still going to code for the same amino acid. And as long as the same amino acid is being produced, we're not going to see a mutation in the phenotype. So this wobble just allows for a little bit of redundancy and protection against mutations, because as long as the same amino acid's being attached, the same protein will be made. Last term to know is rRNA, which stands for ribosomal RNA. And so the ribosomal RNA is going to form a complex with proteins to form the 2 subunits for the ribosome. So we're going to have a small subunit and a large subunit that together form our ribosome. Our completed ribosome will have 3 binding sites, E, P, and A. The P is the site where things start. The P holds the tRNA that carries a growing polypeptide. Here's our P with our growing polypeptide. This right here is the tRNA. The A site is where the next codon or the next tRNA is added with its amino acid, pairing up complementary codon to anticodon. E site is then where the tRNA exits. So this is after its amino acid has been added to the chain, it's going to exit. A ribosome can only hold 2 tRNAs at one time. So now let's look at our steps to translation in a little bit more detail. And we start off with initiation. So here's our mRNA, and the small ribosomal subunit comes in. It's going to bind first to the mRNA so that the first codon, our start codon, is in the proper position. So here's our AUG. This is our start codon. AUG is the start codon in all organisms. All polypeptides are going to start with AUG. So when you're going through translation and practicing, you've got to find that AUG for your start. Then our tRNA with the anticodon UAC, carrying our amino acid methionine or MET, bonds to our first codon. Every polypeptide will always start with MET. Once these 2 have come together, so now we've got our small subunit, our tRNA with the MET. Now our large ribosomal subunit joins, and this first tRNA and amino acid is in the P site. So now we have our initiation complex. Then we can move forward to elongation. So here is that very first tRNA with our first amino acid. Now our next amino acid can come in. We've got codon recognition. They're going to pair up complementary. A polypeptide bond is going to form. This is an energy requiring process. So here we're getting energy from GDP, very similar to ADP, but losing a phosphate, building that peptide bond, and then this amino acid has been removed. Things are going to shift because our chain, our tRNA with the polypeptide chain needs to be in the P site. So we have that translocation. Things are moving. The A site is now available. This tRNA that no longer has an amino acid attached is going to exit. It goes back out into the cytoplasm. Third step then is termination. Elongation continues until all amino acids are attached. Oftentimes proteins are hundreds to thousands of amino acids long. So that process continues until a stop codon is reached. So here we've got our stop codon there in the A site. There are no tRNAs that have the correct anticodons. So we've got 3 codons that say stop. Instead of having a tRNA come in, now we're going to get a release factor that binds with our stop codon. It's going to signal for our polypeptide to be released. The tRNA gets released, and our ribosome comes apart. Our whole complex gets broken down. This mRNA then gets degraded or digested so that the nucleotides can be reused. The free polypeptide then gets folded up into its specific conformation so that it's then a functioning protein.